Welcome back to our designer conference. We've got a couple of sessions this evening, and we're going to start off with Dr. David Minton, and then we're going to have Dr. Danny Faulkner. Dr. David Minton, earlier we gave a pretty good bio for him, a little bit of his background. He is a speaker, writer, and researcher here at the Ministry of Answers in Genesis, a wonderful man, and I want to turn it right over to him to talk about where is evolution going.
Thank you, both of you. Oh my goodness, here we are again. Hope you've all settled down from the meal and everything, ready to... Where is evolution going? I'm not saying I have all the answers, but I've been doing some thinking about it. In fact, I had proposed that this be a kind of a panel discussion. And somehow the message got confused and they made it into a lecture. I'm supposed to give a lecture. I just wanted to talk about it and give a lecture. So I guess this will be a panel discussion where I do all the talking. My kind of panel discussion. So, subtitled on the backs of crystals, because that's kind of a clue to where it's going. First of all, I'd like to begin with God's Word. The fear of the Lord is the beginning of wisdom. Revelation chapter 4, verse 11. You are worthy, O Lord, to receive glory and honor and power. Why? For you created all things, and by your will they exist and were created. Notice that term, will. That means God intended for it. It wasn't just something that happened. Will, that fits in with the word design. That's the whole essence of design. It means knowing in advance what you want to do and then setting out to do it. You are worthy, O Lord, to receive glory and honor and power, and that worthiness is in part due to the fact that He is our Creator. As you know, the world has a very different explanation, don't they? Very, very different. Here's a gentleman, Jacques Monod, very distinguished scientist. Sadly, he died some years ago. He won the Nobel Prize in biochemistry, so we're talking a top-drawer scientist, to say the least. He wrote a book called Chance and Necessity. And in this book, he said that everything in the biosphere, everything in the living world, came into being by just two principles. One of them is chance. I think you all know what he had in mind with that. And the other was necessity. What exactly did he have in mind by that? By necessity, he meant the laws of nature. And that, my friends, is where evolution's going. Chance doesn't get you very far. As we'll see, natural selection doesn't get you very far. You have to look for something that produces sort of self-ordering, something that's a necessity, something that's a law that kind of produces form or structure or order, anything at all to get a leg up on chance. And so in his book, specifically what he said, and by the way, he was an atheist, very outspoken atheist, he said, chance alone is at the source of every innovation of all creation in the biosphere. Pure chance, absolutely free but blind, is at the very root of the stupendous edifice of evolution. I could give you quotes like this. I mean, all evening long, we could just go one quote after another, pure chance. One moral suffice. Julian Huxley, I mean, he's about as major figure as you can find in evolution. In fact, the quote I'm giving you comes from a series of books called Evolution in Action, published back in 1953. Huxley was the keynote speaker when evolutionists were celebrating the 100th anniversary of the publication, of Origin of Species, and the 150th anniversary of Darwin's birth. As you know, just recently, it was the 150th anniversary of the publication of Origin of Species and the 200th of Darwin's birth. So 50 years ago, Julian Huxley was the keynote speaker to represent what evolutionists were thinking about. And this is what he said about evolution. Nowhere in all of its vast extent is there any trace of purpose or even of perspective significance. He said that evolution is impelled from behind by blind physical forces. He went on to say that evolution is I like this expression, evolution is a gigantic and chaotic jazz dance of the particles and radiations. Hey, that'd have been a good place to put a period right there. I mean, it kind of completes the thought. We understand what he meant. But he kept on after this. And I'm telling you, this is just a guess of mine. I do think, maybe, possibly, God came down, grabbed him by the cheeks and says, Julian, you're going to finish this sentence just the way I tell you. Do not deviate one word, because how else can you explain that he went on to say as follows? Remember, impelled by blind physical forces, a chaotic dance of the particles and radiations, in which the only, do you see that word up there, only? In which the only overall tendency we've so far been able to detect is that summarized by the second law of thermodynamics, the tendency to run where? Down. That would be the wrong way if you're on your way from an expanding cloud of hydrogen to, say, Albert Einstein. Somewhere you got to go up a little bit, huh? Up. It's amazing when people say these things. Just to give you further evidence that chance plays a major role in evolution, several years ago I visited the Field Museum of Natural History in Chicago, one of America's great natural history museums, and they actually had incorporated a little gambling casino into their evolution exhibit just to emphasize the role of chance. I took a picture of some kids going through here. I like to think of them as Tommy and Sally. I think they were brothers and sisters. They started off over here at this business on the left here. That's called a dice layout. They have these in gambling casinos, I've been told. And they were having fun just throwing the dice, and banking them off the wall. And I'll tell you, I'm glad that a lot of young people don't read anymore. Because over the top was a sign that said, is life just a game of craps? Craps, that's a slang word for dice, for chance. Think of it. Tommy, your sister Sally there, is she just one big crap game her whole existence? Your mother, your father, your Christianity? Wow, I wonder if the parents prepared them for a challenge like this. Whether their pastor did, or their youth preacher, or their aunts and uncles, or their grandparents. Did anyone prepare them to be challenged with, is your whole existence just one big game of chance? Then they went over to the one-armed bandit, and over the top there was another sign. While they were cranking away on it, on the top it said, over time, tiny mutations add up to big changes. Boy, they got one thing right. Tiny mutations can really add up to big changes. Here's some tiny mutations right here for you. They're cancer cells growing in a dish. They're called HeLa cells. Well, I'll tell you, my wife, Debbie, and I, we both came down with cancer here a couple of years ago. We've been battling away. We've had surgery and radiation and chemotherapy and the whole. It really works some major changes, but I don't think it's what they had in mind. I think they thought or were implying that these kinds of mutations work for the better. Boy, these are some sick cells you're looking at here. Notice the nuclei are all different sizes. There's odd lots of DNA in there. The chromosomes aren't separating properly. These cells came from a lady over 60 years ago from her cervix. She was 31 at the time, checked in in Baltimore at Johns Hopkins, and they took these cells out and have been growing them in dishes ever since. These cells are so out of control, they just keep dividing. Normally the cells in our body only have about 20 divisions, and that's the end, maybe related to our lifespan. These things have been dividing for 60 years, and so far out of her cervix we've got about 20 tons of cells. They've been used in biological research. They're kind of the guinea pigs of cell biology. Dr. Jonas Salk used them to help solve the polio problem, so the lady has certainly served us in that way. Now, I'm talking about, random mutations. I'm talking about just chance. If we had any evolutionists here, they'd say, you're just skipping over the thing that really makes evolution happen. All evolutionists agree that if it was just random change, you wouldn't get anything. But the deal maker is natural selection. In fact, as nearly as I can tell, natural selection does everything for evolution that God does for creation. Well, that better be some sort of a theory. Huh? We're talking a God substitute here. Anybody give me a definition of natural selection? Let's see how close you come. Natural selection is based on changes in the genes, most of which are the result of mutations. We're talking about random mutations. There are mutations in our body that are not random. They're predictable, they're reversible, they're repeatable. They have actually a function, but we're not talking about those. We're talking random mutations. And you cannot select what mutations do not provide. In other words, we talk about selection. Natural selection really isn't selection. I mean, there's nobody out there making choices. What is there to select from? You can only select from the mutations that are already there. You want to do an interesting experiment. Go down to your friendly Midas muffler dealer and order a dozen yellow roses. You won't get them. You know why? They don't stock them. Does it make any difference how badly you need the roses, like you forgot your wedding anniversary or something? No, it doesn't make any difference at all. They don't stock them, you can't select them. So you can't select what's not already there to be selected. Now let's get to what most people would, how they would define natural selection. Survival of the fittest, is that what you've heard? That's kind of what Darwin kicked around, survival of the fittest. Remember now, this is a God substitute, so it has to have a lot of explanatory power. What do we exactly mean by survival of the fittest? Which creatures survive? Why, it's those that are fit. Isn't that true? What precisely do you mean by the word fit? Isn't it the ability to survive? Think this one through for a while now. Which survive the fit? What do you mean by fit? You're able to survive. Which survive the fit? What do you mean by fit? You're able to survive. We're going in circles here. This is called a tautology. And if you like them, I've got another one for you. Do you realize in America today, deafness is a principal cause of total loss of hearing? Think it through for a while. Doesn't really say anything. It's really a poor God substitute, I'll tell you that. Evolutionists have been aware of the problem with natural selection being defined as survival of the fittest for a good many years. In fact, 50, 60 years ago, population geneticists changed the definition. Maybe another 400 or 500 years it'll get down to our schools. But they changed it from survival of the fittest to differential reproduction. What they were saying by that is, look, the individual counts for nothing in evolution. It has everything to do with whether you leave more offspring or not. It has to do with the population. So they redefined natural selection as differential reproduction, simply meaning those that leave the most offspring survive. So all the mutations that occur. Simple question, does this result in you adding to the gene pool by leaving more offspring or not? What about mutations contributing to increased reproductive rates? This is Dr. James Crow, arguably one of America's greatest geneticists. At the time of this picture and quote, he was professor and chairman of genetics at the University of Wisconsin Medical School, and he said that the typical mutation is very mild, usually has no effect, but shows up as a small one. Shows up as a small decrease in viability or fertility. Once again, that'd be the wrong way. By the way, Dr. Crow is retired now, and he's spending what's left of his life working on evolution, trying to improve our understanding of it. Well, let's look at a few more things that are going to impact on this whole business of natural selection to see if it really does work for us. We know it happens, but does it really produce fundamentally new organs and creatures? Moto Kimura is one of the great evolutionary geneticists. He came up with a concept known as Kimura's distribution, or the mutation effect. He said, look, the zero line right in the middle would be a neutral mutation. It would be neither good nor bad, just neutral. The interesting thing, he says, I don't know of any neutral mutations. They're all at least a little bad. So if you look at his curve that he draws, on the left would be the negative mutations, the ones that are harmful, and over on the right, the curve you see over here would be the positive mutations that are downright beneficial. You notice he didn't put anything over there. He said he didn't know of anything that was beneficial to the organism in its normal environment. So even on the negative side, he doesn't quite make it to zero because he doesn't believe there's really any truly neutral ones. But notice that the ones that are not terribly negative are the most numerous. That's a good deal, isn't it? And then as they become more and more negative, there are fewer of them. And those that are positively lethal out here, of course, fortunately, are very rare. But his whole argument is a good part of this curve is in a no selection zone. That is, natural selection has no ability to select out these features, to remove them. And as a result, we accumulate mutations. This is this genetic load that's been building ever since Adam and Eve's kids. They could marry their own brothers and sisters. They had no negative genetic baggage to gang up on them. Today, of course, we can't do that because you carry a double dose of bad genes that become homozygous for something really dreadful. Even if there were just a few good, not very good, but slightly good mutations, they would fall in this no selection zone where you really can't get rid of them, nor can they really help you. There was a mathematician by the name of Haldane, mathematician, geneticist, and he came up with what has come to be known as Haldane's dilemma. He basically argued that for creatures that don't reproduce very quickly, they have longer lifespans, say like humans, primates, what have you, most mammals, you couldn't get enough successful mutations occurring in one individual to really do anything. In a paper he published some years ago, back in 1957, he said, "In this paper I shall try to make quantitative the fairly obvious statement that natural selection cannot occur with great intensity for a number of characters at once unless they all happen to be controlled by the same genes, which is very unlikely." Well, Haldane's dilemma has been sort of dismissed by a lot of people, but in a paper published in the American Journal of Physical Anthropology, this investigator made this rather refreshing comment. Though the scientific community in general no longer regards Haldane's dilemma as a problem, there remains little clarity as to how it became dismissed, and as recently as 1992, the issue is still regarded as a challenge by evolutionary biologists. Well, some people have rather despaired of whether natural selection can really do anything in the way of producing fundamentally new creatures. In a paper in Annual Review of Genomics and Human Genetics, these investigators said, "It remains a mystery how the undirected process of mutation, combined with natural selection, has resulted in the creation of thousands of new proteins with extraordinary, diverse, and well-optimized functions." It's just a flat-out mystery. Now, there's a growing number of investigators and scientists who really are losing their confidence in natural selection as the whole answer. Not too many are willing to throw it out altogether, but they really are not convinced it takes us very far. It doesn't produce fundamentally new structures. It may ensure the survival of a creature and get rid of defective organisms, but as far as making things really new, you can get on the website. This is fascinating if you're not familiar with this website. It's called Descent from Darwin. Notice it's not descent like we descended from Darwin. Dissent, that means disagreement with Darwin. So www.descentfromdarwin.org. What will you see there? Well, if you look over here on the side, a little hard to read up there, so I'll print it out for you. On this website, scientists are signing up to basically agree with this statement. We are skeptical of claims for the ability of random mutation and natural selection to account for the complexity of life. Careful examination of the evidence for Darwinian theory should be encouraged. Man, you could lose your job saying something like that. I'm amazed. If you get on this website, it just goes page after page of scientists, and with relatively few exceptions, I'm not familiar with these people through creation circles. They're not creationists by and large. Last time I checked, there was over 850 scientists that had signed on, at great risk, I can tell you, to their own careers. To sign on, you have to have a PhD or an MD degree and be working in a field of academic science. You can't just be a practicing physician, you have to be engaged in scientific research. The signers include members of the National Academy of Sciences of several countries, and you see many people from top private and public universities and research institutions across the United States and abroad. Check it out. It's kind of eye-opening. Next time you hear somebody say there are no scientists that are at all critical, now, these people probably for the most part believe in evolution. They're just saying natural selection really isn't answering the question for us. Well, a fascinating book was published here a few years ago. It's called The Altenberg 16, an expose of the evolution industry. Susan Mazur believes in evolution. She's certainly not a creationist. But she reported on a rather interesting, almost clandestine meeting that was held in Altenberg, Austria. And this book recognizes or shows that evolutionists recognize the inadequacy of natural selection, at least by itself, to account for evolution. And so 16 very, very influential, very high-powered evolutionists met in Altenburg, Austria, back in 1908. And their whole purpose was discuss the reformation of the theory of evolution. And the symposium was titled, Towards an Extended Evolutionary Synthesis. In other words, we're not throwing natural selection out, but we need more. Now, evolutionists have long said that Darwin didn't necessarily say there was nothing besides natural selection involved. But it's kind of interesting to see, precisely where are they going with this. Before we look at that, I'd like to quickly back up and look at a comment by Dobzhansky that's become almost an axiom in biology. And that is, nothing in biology makes sense except in the light of evolution. Have you heard this before? This is a whole argument for why evolution must be heavily indoctrinated in our schools, because people really bought this idea that if you don't believe in evolution, you can't do science, right? That was Bill Nye's whole thing. Without evolution, you can't do science. In fact, as nearly as I can tell, you can't even be a stand-up comic, which is what part of his career was involved in. How about that? Well, we were just talking about the Altenburg 16. I want to give you a quote from one of the Altenburg 16 people. He happens to be chairman of systems biology, basically cell biology at Harvard University, at the medical school there. He's a member of the National Academy of Sciences. And in an article published in the Boston Globe in 2005, this is what he said, and I quote, "In fact, over the last 100 years, almost all of biology has proceeded independent of evolution, except evolutionary biology itself." Now, if you're going to do that, you need evolution, okay? Now, he's talking about his own field, so he knows what he's talking about. Molecular biology, biochemistry, and physiology have not taken evolution into account at all. Man, that's pretty amazing, huh? Now, don't get the idea he's opposed to evolution. He's a creationist. Far from it. He's part of the Altenburg 16. He's just trying to figure out, how can evolution be made relevant? Evolution was developed, what we call the modern synthesis, about 70 years ago, before we really got seriously involved with cell biology and modern molecular biology and molecular genetics. Is really based on some very old technology. And he's saying, evolution so far doesn't really contribute anything to the modern. He feels that it could. He's not saying that it intrinsically is unable to do that. So where are the Altenburg 16 going? It's kind of confusing. The meeting was sort of closed. There were people reporting on it who managed to get some ideas out of it. I see things going this way. This extended evolutionary synthesis will now extend from natural selection, which is not being discarded, although there were a few there that were ready to pretty much throw it out. One of the fields is epigenetics they're looking at. That is passing on traits outside of the genome. We know there are things going on in the body that are occurring and being passed on to offspring outside the genome. For example, identical twins, which are basically a clone, have different fingerprints. So, something's going on here. Another thing they're looking at is self-assembly. And finally, and this is key, self-organization. Notice they eliminate any possibility of willful design, any input from a creator. Everything has to be spontaneous and natural. It's just they know natural selection doesn't really do it. And so they're looking for intrinsic ordering principles in nature. Ultimately, I believe they'll be going towards something that we could, I think, fairly call occult forces. By that, I don't mean satanic forces. By occult, I mean unknown forces that produce structure. See, this is what the evolutionists haven't come up with, is structure. If you're wondering where we are in molecular biology right now, let's say your goal was to make a men's suit, three-piece, of a certain size, worsted, okay? So far, how far have you advanced on it? You have come up with thread. Now, where do you go with thread to make the three-piece men's suit? You're going to have to weave it into a fabric. You're going to have to cut the fabric pieces to a particular design. You're going to have to sew them all together. We're at the thread level. Genes code for proteins. It's basically like the thread, keratin, collagen, things. But how do you get an ear shape like this? How do you get a feather? Well, I'm not being pessimistic. I think we'll learn more and more about this, but it's going to probably lie outside that little paltry 3 or 4% of the genome that we have a pretty good understanding now, and get into what was once called by evolutionists junk DNA, which is the vast bulk of all the DNA, which every creationist knew would definitely have a function, and of course it does. But this whole idea of life being based in the back of crystals that we heard in that movie is really part of where evolution's going. Because you see, the evolutionist looks at crystals and says, look, here's hydrogen and oxygen, and one would never have been able to determine in advance without actually seeing it, that when these atoms, hydrogen and oxygen, interact together under certain conditions in the atmosphere and changes in temperature and certain humidity and what have you, they can form these crystals. And these crystals, if you notice, all tend to be six-sided or six-cornered. Some of them have absolutely perfect symmetry. Most of them have little defects and are not perfect symmetry. And evolutionists are looking for things like this to produce the order and the structure that's missing just from coding for proteins. Here's that funny line. You've probably all seen this. It's from the movie Expelled, where Ben Stein is talking to Michael Roos, asking about how did living cells begin? Oh, I love this description from Michael Roos.
How do we get from an inorganic world?
Oh, I didn't tell them to hook up the sound. Forgot about that. As you know, he says it probably formed on the backs of crystals. Have you seen this video? Probably formed on the backs of crystals. So Ben Stein says, that's fascinating. Could you expand on that a little bit? And he says, I told you, on the backs of crystals. And basically, he was telling everything he knew, that somehow living things would form on the backs of crystals. Well, what does he mean by that? There is, out of the many, many theories trying to explain how life could have evolved, and technically they're really not theories, one is the clay hypothesis. It's not named after a person called Clay, it's named after clay. Crystals that occur in clay. Back in 1985, Graham Cairn Smith was one of the first to propose that maybe we could begin to get some of the order and complexity necessary for life by having the early organic crystal or organic compounds attached to crystals. And because of the regularity in the crystals, it might begin to impose some order and structure. Moreover, crystals were found to sort of mutate, as it were, to vary a little bit. Not all the crystals are exactly the same. And it was believed that these crystals could pass these changes onto their offspring crystals. Specifically, complex organic molecules were believed to arise gradually on pre-existing non-organic replication platforms of silicate crystals in solution. In 2007, Carr and his colleagues reported that crystals of potassium hydrogen phthalate can act as a source of transferable information. That is, the little changes in the crystal can then go to subsequent crystals. And this is what everybody's getting excited about, that crystals will somehow put things together in an order and produce structure. Well, Carr kind of gave up on the idea when he finally conceded that the transfer of information from one generation to the next really wasn't occurring in a faithful way. Today we have people in the intelligent design movement. I know we can get a little upset with them at times because they don't want to commit to who the creator is. But I think they've at least given us some ideas that we can use. William Dembski in particular has come up with something he calls the explanatory filter. And we can apply that when we look at biological systems or things in nature. Basically, he says you have three possibilities for explaining complexity and order that you see in nature. First, does chance explain it? If chance explains it, then you would expect it to be statistically probable, that it stands a good chance of happening statistically. If you're going to attribute it to chance, you need that. The second possibility is, does design explain it? How do you know if design explains it? He says, is it specified? Is it locked into a particular specification? And third, possibly, does a law of nature explain it? If that's true, it'll be repeatable. It'll just happen again and again as a law of nature. Let's look at these things. First, does chance explain our observations. We might ask, can we distinguish chance from design? Well, let's define our words. I checked the dictionary. Chance is described as the absence of any known reason why an event should turn out one way rather than another. Synonyms are fortune, fate, and luck. Design? Well, no wonder the evolutionists hate the word design. No wonder you can't teach intelligent design in our public schools. Look at the definition from the American College Dictionary. Design means to plan and fashion artistically or skillfully, to intend for a definite purpose, to form or conceive in the mind, to contrive, to plan. I suspect that's how you got to the museum, how you got involved with this conference. You made some plans and came here. Imagine if you would have just left it up to the evolutionary process of chance. You just sort of sit and vibrate in place and see what happens. No, you made specific plans, how you were going to get here, when you were going to leave, how long it was going to take, where you were going to stay, the whole thing. Well, let's look at a couple of rocks, see if we can tell whether they're designed or whether the result of chance. Here's a couple of rocks. What do you think? Either one of them look like they might be a beneficiary of any design. They both have a point at one end, kind of flat at the other. One's a little more symmetrical than the other. Is symmetry an indication of design? Well, how would we tell which of those is designed? Let's look at a bigger piece of rock. You look up on this mountain, you'll see that erosion has caused fissures, ice has broken cracks in it. The just general weathering phenomena has put more wrinkles in that rock than I've got on my face here. When I was up in Canada, some fellow come up to me afterwards and he says, you mentioned you were 75. He says, you didn't look 75 to me. But now that I see you up close, you look 75 all right. I told him I was just about the nicest thing anyone's ever said to me. This is an interesting mountain. 15 years after this picture was taken, random weathering and what have you did this to it. Now, I got a question for you. Let's say you drifted in from another planet. Okay, you don't know anything about presidents. You don't know anything about people. You know a little bit about the natural geology of the planet, but nothing about the biology, and you look up and you see that. Could you tell that was intelligently designed as opposed to the product of weathering, erosion, just natural processes? You actually could. You could crawl up on the nose, for example, of one of the presidents there, and you would notice that whereas normally the fissures and cracks in the rocks follow natural cleavage planes, in this case, the shape of that protruding, what we now know as a nose, people have drilled across with drills and forced the fracture plane to go in planes that are not what you would get if you just left it up to chance. In other words, we have forced the fracture planes, basically a carving process that goes across natural cleavage planes. That's kind of one of the ways we can tell chance from design. What about the basic job of making proteins? That's one of the things we do in our body. We make 100,000 different proteins. Not 100,000 total proteins. Our body has 100,000 different proteins. By the way, when I was in graduate school, there was an old axiom, one gene, one protein. Back then, it was all lockstep. We would have a gene for each protein. So with 100,000 proteins, you'd have at least 100,000 genes that would code for these proteins. How many genes did they come up with when they finally did the sequencing? Well, they were all over the place, but 20, 25,000 pretty well does it. Wow, these genes must be doing some double duty someplace. Huh? It depends on where the reading frame starts or whatever to produce 100,000 different proteins. Proteins are made up of amino acids, and there's 20 different ones that are used. I'll ignore the fact that there are left and right-handed versions. They're all left-handed we work with. So the letters A through T could represent 20 different amino acids. I won't use their real name because it kind of breaks your tongue in two just to pronounce them. Think of the amino acids forming a chain like these beads people wear nowadays that have little messages with different color beads and letters. So A through T would be the full complement of amino acids that we use to make our 100,000 different proteins. How big is an average protein? 400 to 500 amino acids long would be the size of the average protein. So here's an interesting question. I've shown what 500 dots would look like in a row. If we started with an infinite supply of A through T amino acids, and we could just put them in any which way we wanted, so at each position of these 500 dots, you could have any of those 20. My question is, How many proteins could you make that would differ by at least one amino acid? The answer is lots. Lots that can do that. Let's take one of the proteins that's terribly important to us, hemoglobin. Hemoglobin makes blood red. Hemoglobin binds oxygen. You know, if you think about it, our blood is warm, isn't it? You ever drink warm water? If you drink water that's the temperature of your body, it tastes flat. Have you noticed that? We prefer cold water. The reason we like the cold water is more oxygen dissolves in cold water. There's very little oxygen in warm water. Think of it. You have this warm, largely water going through your bloodstream that you expect to carry oxygen down to your big toe. Well, you wouldn't get much down there in warm water were it not for the fact that hemoglobin binds oxygen. It gets down to that cell in the big toe, and then the cell tries to talk the hemoglobin out of it to turn it over, and that involves other enzymes, and it gets very complicated. But here's a red blood cell, and red blood cells are red because of hemoglobin. Iron has a reddish color, just as you see in mineral. How big of a protein is hemoglobin? Well, it's 539 amino acids long, so it's on the larger size as proteins go. And here's an interesting question. If we were to start with the right proportions of all of the amino acids, in other words, the right numbers of A, B, C through T, and we were to just put them in a string like this, we might start just going A through T until we have to change it. How many different combinations would we have to put together before we stumbled upon that combination, which is hemoglobin? Well, years ago, an atheist by the name of Isaac Asimov, of all people, wrote an article called Hemoglobin in the Universe, in which he did the math on this. If you started with the right proportions of all amino acids in our hemoglobin, then how many different ways could you put it together? He called it the hemoglobin number, and there it is. That's a big number. 4 times 10 to the 619th power. That's 4 with 619 zeros after it. That's how many different ways you can put hemoglobin together if you start with the right proportions of all the amino acids. Man alive, how do you tackle a number like this? Let's look at 9 of the zeros, just the red part up there. 4 times 10 to the 9th. No clock has ever ticked that many times. Clocks haven't been around that long. They don't tick that fast. Every time you add a 0, remember, the number gets 10 times bigger each time you add a 0. Let's add a bunch of them. How about 80 zeros? 4 times 10 to the 80th power. How big a number is that? Well, that's a number they throw around as probably the maximum number of atoms in the known universe. That would be a big number. And if we add one more 0, it would be 10 times the number of atoms estimated to exist in the known universe. Well, we could keep going with this. One more batch of numbers, bear with me, 150 zeros. Boy, this is a huge number now. This is called the universal probability bound. Statisticians say that if the chance of something happening is worse than 1 out of that number, the opposite is a certainty. Basically, don't go there. You see, if you want to start playing games by invoking endless periods of time, you undermine the whole statistical foundation of modern science. Statistics means nothing anymore if you're willing to get out the numbers like this, 1 out of this. In fact, statisticians have given us specific warnings not to do this, so they call this the universal probability bound. I don't know how to communicate how big this number is. I guess the closest I can get is to tell you that the whole number up there is so big, it's bigger than the current American national debt in dollars. That ought to impress you. Now, what do we need to make life? We're talking about one protein. By the way, why would an atheist even do this? He did this article because he was showing that we now have a way to sequence proteins back then when he wrote the article. And if we didn't have a way to sequence them, we'd never come up with the right combination by putting them together until they finally worked. By the way, how many out of all those possibilities for putting hemoglobin together, how many are known to work normally? 5. 1 adult hemoglobin and 4 fetal hemoglobins. Fetal hemoglobin has a higher affinity for oxygen, allows the baby to sequester oxygen at a better rate than mom can. In fact, there have been situations where the mother has been asphyxiated or has gone into a lake or river in a car and has died from asphyxiation, but was pregnant, and if they got to her quick enough, they could deliver a living baby because the baby with its fetal hemoglobin was still pulling oxygen out. Well, what's the simplest form of life we know? We've just seen what's the likelihood of coming up with one protein. Well, they say the simplest form of life is Mycoplasma genitalium, and it has 482 protein-coding genes. That would be as simple as it gets, as far as we know. No way, huh? Absolutely no way. Statisticians say this: the statistical explanation must operate in the last instance with very high probabilities. But if our high probabilities are merely low probabilities which have become high because of the immensity of available time, then we must not forget in this way it's possible to explain anything. You just invoke endless time. As they say, it undermines the statistical foundation of modern science. Well, back to our explanatory filter. If chance doesn't explain it, then possibly design explains it. And if design explains it, it will be specified. First of all, what do evolutionists say about design? I mean, I don't know. I look at the hand, it kind of looks designed to me, doesn't it to you? But I know ophthalmologists who say they see no evidence of intelligent design in the eye, and otolaryngologists who don't see evidence of intelligent design in the ear. Well, Richard Dawkins freely concedes that there is evidence for design. I'm sure you're all familiar with Dr. Dawkins, are you? He's a very outspoken atheist. It's been said of Dr. Dawkins that this man knows more things that aren't true than perhaps anyone alive. So here he is. He wrote a book called The Blind Watchmaker, which was subtitled, Why the Evidence of Evolution Reveals a Universe Without Design. So you got the whole idea of the book is that this is going to show you no design, evidence for design in the universe. Then, once again, I think God came down, got a hold of him by the ears and said, Richard, on the first page of your book, you're going to write this sentence, do not deviate. Because on page 1, that would be the first page, he said the following, and here it is printed verbatim, just the way God told him to write it. Biology is the study of complicated things that give the appearance of having been designed for a purpose. Man alive, if I were an atheist writing a book arguing that there is no evidence for design in nature, I would have tried to avoid saying this on the first page. I might have kept it to page 200 or something, or in a footnote, but not on the first page. Well, I agree with him. Biological systems certainly do give the appearance of having been designed for a purpose. Now, how can he say this? The reason he can say this is there's two things about which he's dead certain. Two absolute truths he does not question. Absolute truth number 1 is there are no absolute truths. Absolute truth number 2 is matter, energy, time, and space is the whole of reality. There's nothing outside of the material world. So if you're going to explain the origin of people or cells or whatever, you have to do so in a purely materialistic way. So the fact that biological systems look designed doesn't fool him. He knows there's no designer. So then how does he explain it? He says biological systems counterfeit design. So you see, there again, that's where evolution is going. What kind of counterfeiting mechanisms are there that give the appearance of design that have absolutely nothing to do with intelligence or planning? Well, I agree with him. Biological systems give the appearance of being designed for a purpose. One organ I studied, as I mentioned earlier today, is the placenta. What an amazing organ that is. Here is a placenta. You can see on the left the side that the fetus would see. The other side is the side that's plugged into the mother. And when the placenta is unplugged from the uterine wall at the time of birth, it severs several arteries that supply the blood to the placenta. In fact, the blood flow into the placenta is so fast at the time that the baby is born that approximately all of the blood of the mother goes through the placenta in 10 minutes. This means that when the placenta unplugs, you are severing several arteries, a little smaller in diameter than soda straws, that are bleeding so fast that in 10 minutes you will totally exsanguinate the mother. Anyone see a problem here? Could any wound be worse than this? It's been said this is the worst wound anyone gets and lives. What would a surgeon do in a situation like this? Well, she'd probably grab a bunch of hemostats and just start clamping blood vessels as fast as you can go. Well, that's what God does too, except he has the hemostats already in place. Little muscular sphincters in every one of the arteries and veins supplying the placenta. As soon as the placenta unplugs, the dotted line means it always tears on the side away from the sphincter, so the sphincter stays with the mother. The muscles clamp down. Instead of being insanguinated in 10 minutes, maybe lose a cup of blood in a normal pregnancy. How's dumb luck working for you so far? So anyway, the placenta has unplugged, and if you look at it, the side that was plugged into the uterus is lumpy. And those lumps, of which here is one lump, they're called cotyledons. And these approximately 20 cotyledons look like little trees. If you look in a diagram, for example, between the two blue lines there, this would be one of the cotyledons, approximately 20, look like a little lump up here. This is the trunk of the tree. You have one trunk, you have lots of branches. The branches go out to twigs. Some of the twigs attach to the uterine wall are called anchoring villi. Most of them are free. Baby's blood runs inside of this little tree, like the sap. Mother's blood runs through the branches of the trees out here, so it's like a little forest of 20 trees. Mother's blood is like the wind blowing through the trees. Baby's blood is like the sap inside the tree. So baby's blood is separated from mother's blood by basically what you might call the bark of the tree, the surface. Let's look at that. One of those little tips right there, see, one of the little twigs? We'll look at that in the microscope. We've cut across it, sliced it so we're seeing it end on. And when we look at it up there, this would be baby's blood inside the twig. And this red blood cell, wants to jump ahead on me. The red blood cell just outside right here, that would be mother's red blood cell. Over to the right here would be a white blood cell from mother. Inside here is a white blood cell of the baby. Do you see how close the blood is? But it's separated by this pink line that you see right here. Now that pink line is a cell. Did you hear me right? Singular, a cell. It forms from millions of separate cells that fuse together. They're called cytotrophoblast cells. They fuse, and when they fuse, they form the largest cell in the body. It's believed that the surface of the placenta, the pink line you see going around the twig, cut in cross-section, which is called a syncytial trophoblast, is one cell. It's a syncytial cell having been formed from millions of separate cells that fuse. So lots of nuclei, but basically one membrane. How big would this cell be if you could flatten it out instead of covering all these little trees? Estimated to be the size of a living room area rug, 8 by 11 feet. Mother's blood's on one side, baby's blood's on the other. Everything that goes from mom out here to baby or baby to mom has to go across that seamless surface. Water goes across passively. Unfortunately, alcohol goes across, so if mom consumes a lot of alcohol, the baby could be born with fetal alcohol syndrome. Here and there you see little clusters of nuclei. That's where the nuclei are kind of pushed out of the way, so there can be lots of things going across the placenta. Why, in addition to this, do I think that the placenta shows evidence of design? Because it does everything. I mean, it accomplishes the function of most of the organs in the body. It's a lung, it's a liver, it's a digestive system, an endocrine system, a urinary system. Baby can be born with no lungs. Do okay till you take the placenta away, then it dies. You know those 100,000 proteins I told you about in the body? There's one of them you maybe haven't heard of. It's called transferrin. Without transferrin, none of us would be here. How about that for important, huh? You see, the baby has to make its own blood up here, and the baby's only source of iron is from mother's blood. It comes from mother's blood up here. Only source of iron has to come across to baby's blood. Problem is, the placenta is impermeable to iron. Anyone see a problem here? The baby can't make blood. The blood is the life. Iron doesn't come across the placental barrier, except for transferrin. Transferrin is one of a couple of hundred transport proteins in the placenta that go across, pick up things from mother's blood, bring it across the barrier, and release it to baby's blood. Man, I'd like to play a little game with the evolutionists and say, look, I'm going to give you a head start. I'm giving you everything in the entire cosmos as a head start. Man, that's generous, huh? I'm just keeping back one thing, transferrin. I'm putting that in my pocket here. I keep this, you get everything else. You're done. All placental mammals, you'd be the last generation on Earth. Now, you could go to the gods of evolution and say, this rascal Menton, he gave me everything, but he held back transferrin. Could you give me a few million years to dumb luck my way into transferrin? Nope, one generation, you're done. Man, it isn't so much irreducible complexity as is insurpassable complexity. You do not get to go to step C without B. Sort of like Monopoly, do not go to jail, go directly to jail. Do not pass goal. Well, back to our explanatory filter, does a law explain it? Now, this is interesting. This is a place evolutionists are looking where there could be laws that would give the appearance of design. Several years ago, old Charlie Darwin had a friend called Asa Gray. Asa Gray was a scientist. He was not a Christian, and he wasn't entirely convinced Darwin had an explanation for things, and so he just bugged him all the time. And one of the things he bothered him with is, what about the bee's honeycomb, Charles? That's the least amount of wax you can use to enclose the maximum amount of honey. Does your theory explain that? Well, there's what the honeycomb look like, perfect hexagons. Can't you just see the bees out there with the little protractors and everything getting 120-degree angles at the corners? Well, Darwin wrote a letter back to Asa Gray. In the letter, he says, your letter actually turned me sick with panic. Boy, Asa Gray really spun his crank, didn't he? He did remember the peacock tail? That was Asa Gray again. Man alive, he didn't let up on him. Darwin went on to say that this is the most wonderful of instincts. Beyond this stage of perfection in architecture, natural selection could not lead. For the comb of the hive bee, as far as we can see, is absolutely perfect in economizing labor to wax. In other words, he said that hexagonal arrangement, that's as far as natural selection could go. That just takes it to the top. Is that true? Take a look at this picture right here. Those are soap bubbles floating in water. A single layer of soap bubbles. My lab tech called it a unibubular layer. Those bubbles form hexagons spontaneously. This is a law of nature. It is a law of close packing. You take a bunch of pennies, put them on a table, push them as tight as they can go. Each penny will be surrounded and touched by 6 pennies. Not 7, not 5. And if you could squeeze them together till you squeezed out all the spaces, you get hexagons. So you see, you don't need an enzyme for this, you don't need a protein for this. This is a law of nature, a law of hexagonal close packing. This is kind of where evolution is going. Can we find more things like that can give us some order, that can give us some structure? So Darwin got bamboozled. By the way, a solitary wasp just makes one cell, and it's a cylinder. Basically do this, you make a cylinder around yourself. But you get a bunch of bees doing this together, and those cells pack against one another like the soap bubbles. You get a hexagon. Hexagons are widespread in nature. For example, covering the organs of our body, if you open up the body and look inside, is like a drape. It's called the mesothelium, or the mesentery. And if you outline the cells of the mesentery, they're hexagonal. Lining of blood vessels are hexagonal. Lining of the heart is hexagonal. All simple epithelia, ultimately packed together, is hexagonal. It's a law of nature. Now, we're talking 2 dimensions, aren't we? Just 2 dimensions. Basically, circles, when pushed together in 2 dimensions, become hexagons. Here's an interesting question for you. If circles become hexagons, what do spheres become? Now, as we go into 3-dimensional space, what happens there? That wonderful creationist, Lord Kelvin, figured this out about 130 years ago. It forms a structure called the orthic tetrachedecahedron. It's a 14-sided polygon. This represents essentially the minimum partition of 3-dimensional space. Soap bubbles packed in a froth, and there was more than one layer, within the interior of the froth will approximate that shape. Notice that, at least oriented one way, they form columns. These columns go vertically up through. Well, you look at pith in the stem of a plant, in this case in an elder plant. The longitudinal axis through the plant, you can see we have these hexagonal structures that are arranged in columns. This is this close packing phenomenon that produces these columns of cells and pith. If you look at a cross-section of the stem and look down the columns, you see, what you have on the right, hexagons going down the column. Now here's a little interesting side story I want to share with you. Back in 1665, Robert Hooke designed one of the very first microscopes that was used. And one of the things he looked at with his microscope was cork, like bottle cork, which is really plant cambium from a plant. And he noticed that in the cork, the cells were in a row. That was this close packing phenomenon again. In fact, in the scanning electron microscope, I took a picture of what Hooke was seeing, only I had a lot better resolution than he had. And he saw these little boxes in continuous long rows. And he thought that they looked like the rooms that monks live in, in a monastery. They're called cells. And that's how we got the name cell, because of the cells in a row, which is related to this close packing phenomenon. Well, I was aware of all that, and so years ago we were approaching a problem in skin, and that's the dead layer on the surface of the skin where you see the yellow area there. I was interested in how are the cells arranged. There must be some kind of close packing phenomena there, but we couldn't quite make out positions of the cells. So with a special technique I won't get into, we swelled the cells up, and we did, lo and behold, they're arranged in columns, just like poker chips in a casino, I've been told. Not only that, but if you look at the edges there where the cells in adjacent columns meet, they interleave perfectly. I never saw two together. They were always just like this, perfectly interleaved. In fact, if we take the area in the yellow box and we blow it up in the electron microscope, transmission electron microscope, we notice that here is a cell coming in from the right. It comes, oops, very touchy here. A cell coming in from the right, then one from the left, one from the right, one from the left, one from the right. At this magnification, the cells would be a block off that way, the other edge. But here where they're interleaving, it's a perfect regular interleave. These are the dead cells in our stratum corneum of our skin. And it dawned on me, this is probably a close packing phenomenon. So in studying it, lo and behold, that was the case. If you looked at the columns edge on, they're in perfect stacks. If you look down the top, they're hexagonal. And I was able to demonstrate that basically a truncated form of this close packing structure, the orthic tetrachydecahedron, accounted for the column architecture. And there was evidence for that when we looked at the cells in the scanning scope. Notice that this particular polygon has alternating four-sided and six-sided faces. And that's what we saw looking at the overlapped edges of the cells in the skin. So here is a spontaneous phenomena. It is a law of nature. It produces this seemingly marvelous column structure. And evolutionists are going to be looking for this kind of thing to try to explain how we manage to get structure and order. But boy, have they got their work cut out for them. Let's just analyze this. I fly in airplanes quite a bit, and the other day when I was looking out the plane, there was nothing down there but air. And I wondered, what keeps this thing up in the air? Is it a flying carpet or what? Then it dawned on me, man alive, I was sitting in the middle of 6 million non-flying parts. I got on the internet and asked, how many parts in a Boeing 747? It said 6 million, mostly rivets. And I thought, there isn't a part in this plane that flies. What makes the plane fly? What would I need to put this thing together? None of those parts fly, but what would I need if I wanted to assemble it and get a flying plane? Instruction manuals, right? We call all those parts complex. You can't fly complexity. Have you heard people say, how can you believe the eye form by chance is too complex? That's not the issue. You've just said, how can you believe the eye form by chance? Sure, a lot of parts in the eye. Hey, there's a lot of parts on the runway. You can't fly it. Complexity is not the issue. You see, you'd get the manuals, and once you had the manuals, we call that specified complexity. Remember, that was one of the criteria for a law of nature, is it specified. Specified complexity means that you shall take the parts and put them together as follows. Do not deviate. Part A goes with B down the line. Once you get it assembled, why is it able to fly? It's able to fly due to integrated complexity. That is, the parts work together in concert. fact, that's a good name for it. I like to go to the symphony here in Cincinnati. I go early to hear them tune up. Have you heard a symphony tune up? Worst sound you ever hear. Squeak, squawk, rumble, bang, tinkle. I mean, everything's going on at once. Then the conductor comes out. Everybody opens a score. Specification, the score. All open to the same page. Conductor sets tempo, dynamics, specification. They now all play together, and it sounds beautiful. And see, right there, we went from the tune-up, which is mere complexity, to Beethoven's Fifth Symphony, which is specified and integrated complexity. The instruments are playing together in the orchestra. So the difference between order and complexity is also important. Some people think life's about order. That's why the evolutionists are getting excited about crystals. We can get an order. Hey, order isn't what it's about. Here's a couple of fences here. One neighbor puts up the fence in the top. The other neighbor puts the fence in the bottom. Don't you worry about him? Which of those two fences has the most order? The top one. Which is the most complex? The bottom one. A good way to know that is, what's the smallest set of instructions you could write for the upper fence? You say, start with a stack of short boards and a stack of tall boards, put up a short one, put up a tall one, continue in this fashion until you run out of boards. What about the lower fence? Oh my goodness, where do we start? There are boards missing, there are short ones together, long ones together. But while it's complex, what's our question? Is it specified or integrated complexity? Bingo! Your neighbor's trying to communicate with you. He's using International Morse code. For the dashes, or the dots, he's using short boards, and for the dashes, a long board. So that would be did it, da dit, da dit, da da da, da dit, on down the line. Wow, the neighbor has to know what language you speak, English, French, German. If it was more than one word, he'd need syntax and grammar. And here we've spelled information. You see, the issue is not order. That's not going to get you anywhere. In fact, death is more like order. A crystallization, a minimum free energy state where you don't go any further. No, you need the complexity, but it can't be just complexity. It has to be specified and integrated. And that's what we have in our genetic code. We have a coding system just like Morse code. And by the way, nobody's ever seen a coding system come into existence by chance. All codes are the product of an intelligence. And that code codes for our proteins and gets them all in the right place. And what do the evolutionists say about that? Well, here's Professor Paul Davies, expert on the origin of life. So if you're going to find out how the life has evolved, he's going to tell you. How did stupid atoms spontaneously write their own software, and where did the peculiar form of information needed to get the first living cell up and running come from? Definitive answer up there. Nobody knows. One person knows. There is a book. And in this book, we read in Luke chapter 1, verse 37, for nothing is impossible with God. If we go to Hebrews chapter 3, verse 4, we get a verse that's so beautifully simple, I can understand it. It says, for every house is built by someone, but he who built all things is God. Why don't people believe that, huh? How can ophthalmologists not see design in the eye and otolaryngologists not see it in the ear? This is why. Proverbs chapter 20, verse 12, the hearing ear and the seeing eye, the Lord has made both of them. No guesswork there. The Lord just says straight away, see the eye and the ear, I made them. What's the problem with that? Here's the problem with that. Psalm 94, verse 9, this is scary if you're an unbeliever. He that planted the ear, shall he not hear? He that formed the eye, shall he not see? How well you figure the creator of the ear can hear our thoughts. How well do you figure the creator of the eye can see into our hearts? This is the same creator who once said, you shall be perfect as I, the Lord, thy God, am perfect. How are you folks doing on the perfection scale? I'm coming up really short. To be brutally honest, I'm the worst sinner I know personally. Not going to speak for anyone else. How can I face up to a God who can look into my heart, look into my thoughts? You want the short answer? I can't. I'd run from such a God. You couldn't drag me into this place with chains if that was the end of the story. But he's not just my creator. Most importantly of all, he's my Savior. And that changes everything. Loved me enough to come into this world to take my sins and pile them all on him. He took the hit I should have taken. And wow, once you know that, Isn't it great to have a hearing and seeing God? How about that for our concluding verse, Psalm 34, verse 15? The eyes of the Lord are upon the righteous, and his ears are open unto their cry. Thank you.